Chapter 9, Energy Metabolism, Screencast 2. This screencast will be focusing on how ATP is produced from carbohydrate. And one of the things that I want us to think about here is we want to think of carbohydrate and glucose as the gateway molecule for metabolism. So here we will be introducing the majority of the pathways that are going to be used to produce ATP aerobically. So what we'll find out later when we start looking at how we get energy from protein and lipids, there's not going to be a lot of different stuff going on. There may be a little bit, but not anything that's going to be really overwhelmingly different. So it will utilize the pathways that we are introducing here. The other thing that I want you to remember here is when we're talking about the oxidation of glucose, we want to remember that it requires oxygen, and that's what cellular respiration is. It is the process in which our cells generate ATP. The food that we consume are oxidized to form ATP, and we want to think of oxygen as the final electron acceptor. Now, these processes may be aerobic or anaerobic. And aerobic is when energy or oxygen is readily available. It's much more effective. And generally, we're going to see a net gain of 30 to 32 ATP. Don't get caught up in the numbers, okay? Anaerobic, when oxygen is not adequate or present, then we're only going to get a net gain of 2 ATP. This is through the process of lactate formation. And keep in mind, 2 ATP is better than no ATP. Now, there are a total of 4 different stages involved in the aerobic carbohydrate metabolism. And so the first one that we want to look at is glycolysis. We will be starting with glucose. And remember, glucose has 6 carbons, and we will end up with 2 pyruvate. Each of those is a 3 carbon. Now, notice this takes place outside of the mitochondria. Then there's going to be a transition reaction in which pyruvate will be converted into acetyl-CoA. The third stage is going to be the citric acid cycle or the Krebs cycle. And then we'll actually have the electron transport chain. And this is where the majority of ATP is actually produced aerobically. Notice glycolysis does not take place within the mitochondria, yet the transition reaction, the citric acid cycle, and electron transport chain all occur within the mitochondria. That shouldn't be a surprise since we consider the mitochondria to be the cellular powerhouse. Now, the first stage is glycolysis. And what's going to happen here, basically, glucose will be oxidized to form 2 molecules of pyruvate. We will consider that to be an intermediate because it's not carbon dioxide, water, or ATP. We'll also get some electron transport molecules, and that is NADH plus the corresponding hydrogen ion. Keep in mind how important this is, because we're going to come back to that, and these are the electron transport molecules, and we'll be using them a little bit later when we start thinking about the electron transport chain. There is a net production of 2 ATP. That's also important to remember here, okay? And so this is the beginning of energy generation. Now, the important thing to remember here is this process of glycolysis occurs within the cytosol. And the role here is basically to break down carbohydrates to generate energy and produce building blocks for other compounds. Glycolysis does not require energy, but one thing that does make glycolysis different is you actually have to invest 2 ATP. And that is why we have a net gain of 2 ATP, because you have to pay back the 2 that you originally invested in overall. Now, glycolysis, once again, is an elaborate 10-step pathway. So lots of reactions that are going on here. And basically what we're going to end up with is 2 pyruvate and 2 NADH plus hydrogen ions. Then we have the transition reaction, and this is how pyruvate is going to be converted into acetyl-CoA. So this also is going to result in a product, because notice pyruvate is 3 carbons, but acetyl-CoA is 2 carbons. We just don't lose carbons, and you'll notice that CO2 will be one of the things that is actually produced, and we will consider that to be a product because it is carbon dioxide. There will also be the formation of NADH and hydrogen ion, And remember, we've got to double that because we actually have 2 pyruvates overall. Now, once this takes place in the mitochondria, it requires oxygen. And once we get to acetyl-CoA, we are at a point where it is irreversible. So we cannot go backwards from acetyl-CoA. The third stage is the citric acid cycle, and we are going to actually look at acetyl-CoA as the beginning of the cycle. So to begin the citric acid cycle, the 2-carbon compound acetyl-CoA combines with a 4-carbon compound oxoacetate to form the 6-carbon compound citrate. In the process, the corresponding CoA molecule is released and can be reused. So keep in mind we're back to that magic number 6. And notice we eventually have to get back to oxoacetate. The 6-carbon citrate is oxidized, forming the 5-carbon compound alpha-ketoglutarate, NADH plus hydrogen ion, and carbon dioxide. So we went to 6 to 5, so that gives us another lost carbon and a carbon dioxide. The 5-carbon alpha-ketoglutarate is oxidized, forming the 4-carbon compound succinate, NADH plus hydrogen and CO2 and guanosine triphosphate, which eventually will be ATP. We're going to think of this as an ATP, not GTP. The 4-carbon succinate is oxidized to the 4-carbon compound fumarate, and this is where we actually get FADH2. Then the 4-carbon fumarate is oxidized, forming the 4-carbon compound oxoacetate, the compound used to actually begin the citric acid cycle, step 1, and another NADH. So we are not generating a lot of ATP here. We're looking at basically 2 ATP produced, but remember, the citric acid cycle will spin twice per glucose. And so we will get a lot of electron transport molecules, a total of 6 NADH and corresponding hydrogen ions, and also 2 FADH2s, and not a big producer of ATP. So let's really think of this as the job of the citric acid cycle is really the generation of electron transport molecules. Now, we do have products. So once again, we have carbon dioxide, and we also have some ATP, not a lot, but we now have addressed all of the carbon dioxide that we expect to be produced from the oxidation of glucose in the presence of adequate oxygen. But please keep in mind, the citric acid cycle does not require oxygen. The last step or stage is the electron transport chain. And there are a multiple of processes that are going on simultaneously. One we have to think of is oxidative phosphorylation. So the energy derived from our electron transport molecules, NADH plus the corresponding hydrogen ion, and FADH2 is actually used to form ATP. The NADH and hydrogen ion is oxidized to NADH. FADH2 is oxidized to FAD. This process requires copper and iron. Iron is a major component of the cytochromes. Oxygen is used and combines with hydrogen and electrons to form water. The majority of all ATP is actually generated in the electron transport chain. And so we can see this here as far as what's going on. Now, one thing that I want to keep in mind here is for every NADH and hydrogen ion we produced along the way, we will actually get a total of 3 ATP. We can see that here. Now, if we look at FADH2, notice it enters the electron transport chain at a different place. So it's only going to pick up 2, or ends up in yielding 2 ATP for ADH2. So the role of oxygen here is the final electron acceptor, and we actually see the formation of water. Now, we want to describe oxygen as the final electron acceptor. So oxygen is essential for the metabolism energy to continue. Without it, metabolism will stop and death will take place. It is the final acceptor of electrons and hydrogen ions generated from the breakdown of energy-yielding nutrients. And basically, cells rely on this to extract enough energy from energy-yielding nutrients. Now, anaerobic metabolism takes place when cells do not have mitochondria or oxygen is in not large quantity. Pyruvate will be produced during glycolysis and converted into lactate. The conversion of pyruvate to lactate involves this enzyme, pyruvate dehydrogenase. It's going to move hydrogen from hydrogen ion and hydrogen ion to pyruvate. This is going to create NAD plus and lactate. Here we can actually see this process. And once again, basically it's a stopgap measure at the cellular level to maintain the production of some ATP in the absence of oxygen. This basically will end up with a net of 2 ATP. And keep in mind, 2 ATP is better than no ATP, because what happened here is oxygen is no longer available and you're no longer having NAD+, and you need that to get this whole process going and to accept more electrons. So without that, we're stuck with glycolysis, which takes place outside of the mitochondria, so it does not yield in a lot of ATP, but we get, once again, 2 ATP better than no ATP. Now, the Cori cycle, this is something that takes place during high-intensity exercise, and muscle cells are relying on anaerobic respiration for the production of ATP. This causes lactate buildup and NAD plus regeneration. Lactate is transferred to liver, will be converted back to glucose. So let's take a look here. So in the absence of oxygen, muscle produces lactate from pyruvate. We do not want to think of lactate as an endpoint. Lactate actually leaves the muscle via the blood and enters the liver. Liver enzymes actually convert lactate to glucose through the use of ATP, and glucose returns to muscle where it can actually be used for energy.